synergistic connectome of Neuropeptides elegans. So please start.
Hello, so my name is Lidia Ripoll Sanchez, and I'm a graduate student at the Schaefer and Vertus lab in the MRC Laboratory of Molecular Biology in Cambridge. And I would like to thank the committee from the WORM meeting for giving me the chance to present here today. So when we touch a worm, it moves backwards. Even a straightforward behavior like this one is ingrained in the structure of the nervous system of C. elegans. So if we want to fully understand this behavior, we need to fully understand the structure of this nervous system. For the past 35 years, we've had the full connectome of C. elegans that gives information about the gap junctions and synapses that link the individual neurons in this connectome. However, in all these years, we still have not been able to fully functionally understand and interpret this connectome. Besides gap junctions and chemical synapses, neurons in C. elegans communicate through other means, which are usually extrasynaptic communication. One hypothesis why we still cannot fully understand this connectome is that we are not considering this extrasynaptic signaling. Now, previous research in C. elegans has shown that this extrasynaptic signaling is fundamental to understand some behaviors. For example, in locomotion in response to food presence, key signaling events happen extrasynaptically in between neurons that are not synaptically connected, such as the AVK connection to VC neurons happens through the extrasynaptic release of FLP1 neuropeptide and its binding to the NPR6 receptor in VC neurons. In arousal, we see a similar situation where mechanosensory neurons connect to RID through the extrasynaptic release of FLP20 neuropeptide and its binding to the FRPR3 receptor. And in learning, we see the connection between ASG and AFD neurons through the extrasynaptic release of NLP44 neuropeptide and its binding to NMER1. Now, given that we know that all these extrasynaptic connections exist, we can infer that they form a network. And then based on the different modes of communication between neurons in the C. elegans connectome, we can split this multiplex network in several layers. The first layer would be a synaptic layer, and then a gap junction layer. These two layers require physical contact between the neurons for communication to exist. So we call these wired connections. And then we can find connections through neuropeptide GPCR interactions or through small molecules like monoamines and other interactions with GPCRs that do not require physical contact between the neurons and happen through diffusion of the molecules in the space. These are wireless connections. In this project, we want to understand better how these wireless connections affect the overall C. elegans connectome through mapping and analyzing the neuropeptide GPCR network. Now, how do we build this network? Well, the first thing that we need are to identify the neuropeptide receptor couples. For example, the NLP44 neuropeptide binds to the NMER1 GPCR. Then we need to identify which individual neurons express each neuropeptide and each receptor. In this case, ASG neurons express NLP44 neuropeptide, and AFD neurons express NMER1 GPCR. Then with this information, we can build an extrasynaptic link between these two neurons, which becomes the edge in our network. Now, in order to get these neuropeptide receptor couples, our colleagues from the BEADS lab in Leuven performed an in vitro reverse pharmacology assay, and they identified a wide range of neuropeptide GPCR interactions. However, a lot of these interactions happen with different affinities. So in order to get the interactions that happen with the strongest affinity in vivo, we decided to threshold this dataset for an EC50 of 500 nanomolars, which yields us 93 individual neuropeptide GPCR interactions. Then, to identify the neurons that express each neuropeptide and each GPCR, our colleagues from the CENGEN project identify single-cell RNA-seq expression for each neuron in C. elegans. Again, this dataset has different gene expression levels for the same gene in different neurons. So in order to get the highest rate of true positives, they decided to threshold this dataset based on reporter strains. With these two datasets, we get a large number of connections in the worm, And these connections can happen at different scales depending on how far the neurons they connect are in the anatomical structure of the worm. So in order to get the connections that we were more comfortable with, we decided to spatially constrain the network and build 3 different models depending on these different scales of connections. Our long-range model includes all possible neuropeptidergic connections in the worm, so it connects all neurons and has no spatial restrictions. Our middle-range model connects only neuronal processes that are in the same anatomical areas of the worm, like the head, the tail, or the mid-body. And our short-range model only connects neurons that are in the same process bundle. This short-range model is the one that we've decided to mainly continue forward with the analysis, since a lot of these short-range connections have been experimentally proven to exist in vivo. Furthermore, if we look at the number of connections of each type in the overall system, more than 50% of the neuropeptidergic connections in the system are short-range connections, whilst the mid and long-range connections are a much lower percentage. If we compare these short-range connections with the number of connections that we find in the synaptic and gap junction systems, we see that there's more than 10 times more neuropeptide connections between the neurons in the system than synaptic and gap junction connections. Now, the short-range model is built of these 93 individual neuropeptide interaction pairs. And each of these neuropeptide interaction pairs can form an individual network that has a particular topology. The topologies of these networks are dependent on the individual neuropeptide receptor pairing. So even the networks that use the same neuropeptide or the same receptor, like the ones highlighted here that use all the same receptors, have individual different topologies depending on the neuropeptide they bind. In the in vivo system, all these networks appear together. So if we want to study this system as closely as we would find it in vivo, we can group them all into an overall neuropeptide network, where most of the connections happen between the sensory neurons, interneurons, and motor neurons, and the firing system is a bit on the side with a less number of connections connecting it to the rest of the system. Now, what else can we learn from this neuropeptide network? From a first glance, we can see that it's a very dense network. There's a high number of connections in between the neurons in this system. Then we can also see that these connections can happen at variable weights. So the number of neuropeptide GPCR pairs that connect 2 neurons in this system is very variable. For example, the connection between PQR and AVDL neurons happens with 18 different neuropeptide receptor pairs. As well, we can see that a lot of neurons in this system express the ligand and the receptor for the same neuropeptide receptor pair. So we can say they have self-connections. They can connect with themselves. We can look more at these individual connections, looking at the degree distribution of the network. We define degree as the total number of incoming and outgoing connections that a neuron receives in the system. And the neurons with the highest degree are defined as hubs. If we look at the neuropeptide degree distribution, we see that there's a high amount of hubs that have a high number of incoming and outgoing neuropeptidergic connections. Besides, a lot of these hubs are neurons that don't have a high synaptic connectivity. And among those hubs, we can include AVK neurons, PVT, PQ neurons, and PVR. If we compare this degree distribution with the synaptic degree distribution, we can see that the hubs have 5 times more neuropeptidergic connections than synaptic connections. Besides, the theoretical maximum for this network is to have a degree of 604. That means that every neuron would connect to every other neuron. The hubs in this network have 500 connections, which means that some of these hubs connect to pretty much every other neuron in the system. If we correlate these 2 degree distributions, we can see that the synaptic hubs, so the neurons that have the highest synaptic connectivity, have also a high neuropeptide degree. So they have a very high neuropeptide connectivity. But not all neuropeptide hubs have high degree connectivity. This group of PVR, PVT, PVQ, and AVQ neurons AVK neurons, sorry, have a high neuropeptide degree, but don't have a high synaptic degree. So we could say that they are neuropeptidergically specialized neurons. Another way of looking closer at the interactions between these hubs is to look for a ridge club. So we consider that a network has a ridge club if the hubs in this network are more connected between themselves than to the rest of the network. In this case, the C. elegans neuropeptidergic network does have a ridge club of about 168 different neurons, in which we include the ones that we mentioned before, so PVT, PVR, AVK, and PVQ. Now, previous research linked the synaptic ridge club to individual behavioral states. In this case, the synaptic ridge club neurons are recruited to motor command samples. Our hypothesis is that these 168 neuropeptidergic neurons could also be recruited to command certain global states in the world. And that specifically, some of these neurons, the highest degree hubs, have already been identified to some behaviors that we could consider global. So for example, AVK has been linked to arousal and navigation behaviors. PVR has been predicted to control motor neurons. And PVT has been linked to arousal. In the future, we want to continue analyzing the connections between these neurons, study their dynamics in time, and look more at the individual neuropeptide receptor pairs that they use to communicate with each other. So to summarize, we've built a map of the neuropeptidergic connections in C. elegans. It forms a dense network with a large coordination center that includes every neuron in the C. elegans nervous system. And some fundamental neurons in this neuropeptidergic network are not fundamental in the synaptic network, but are specialized neuropeptidergic networks that could have important command roles. With this, I would like to thank everyone involved in this project. And I would like to invite you all to attend the posters from the Schaefer and Beats Labs. And I'm happy to take any questions.
So thank you, Lydia. So when looking at neuropeptide for mapping, do you use prohormones or processed?
So because we are working with mRNA data, we are working with neuropeptide precursor genes.
Does pharmacological analysis data show the neuropeptide binding to multiple GPCR and the vice versa?
Yes. So, sorry, I'm gonna go back to this slide. I think it shows it a bit better. So here, this pharmacological analysis shows that some GPCRs can bind several neuropeptides. So in this case, there's some that bind up to 11 different neuropeptides, and there's also some neuropeptides that can bind different receptors. So it adds this extra level of complexity to the network.
Okay, thank you, Lydia.